So we're going to start off with ATP, which is an old friend of ours. We know it's a nucleotide with a 5-carbon pentose sugar and an adenine nitrogenous base, and then 3 phosphate groups. So we've seen this molecule before. We're going to look a little bit more about, just a reminder about the sort of recycling or recycled, rechargeable nature of ATP, and also a little bit about how we actually transfer or use the energy that's trapped in those high energy bonds. We've seen this graphic before when we were looking at enzymes. We also saw this graphic when we were looking at energy at the beginning of this unit. So this is pretty familiar to us. Should note that this one on the y-axis, or sorry, on the y-axis is measuring free energy, or delta G, Gibbs free energy, which we discussed in a previous PowerPoint. And on the X portion of the graph, we're measuring the progress of some kind of reaction. And we're looking at 2 possibilities here. The red one would be the reaction without an enzyme, and the black one would be the same reaction with an enzyme to lower the activation energy. And we can see the change in free energy is the same regardless. And this kind of reactant, we can see the, reaction, we can see the reactants have more energy than the products. So we're looking at energy being released, an exergonic type of reaction. So again, in the words, basically the same sort of idea, talking about activation energy and spontaneity of reactions and the role that enzyme plays in that. Okay, big red letters, so must be important, right? In cells, energy from catabolic reactions is used to power anabolic reactions. So this idea of coupled reactions, and we did see it in a PowerPoint before, but this idea that we couple reactions, so things that are losing energy, or reactions that are losing energy as they're occurring, that energy that's being lost there is going to be used to power some other reaction that's happening in the cell. And the classic example of that, of course, would be the breakdown of glucose being used to power other cellular work that's occurring. And the intermediate in that case is usually ATP for us. So when we break down glucose, we are going to create some ATP from that. So the breakdown of glucose is, for instance, going to be able to power the maybe the building up of something like a polypeptide chain, or a storage polysaccharide, or something like that that requires energy to occur. And again with the red letters. Again, the idea of coupling the hydrolysis of the ATP to supply that energy. So again, we did see this already, and I think this graphic, is one that's familiar to you. In fact, it's the same graphic we looked at before, where we've got glucose on the left-hand side being broken into carbon dioxide and water, so high energy molecule to low energy molecule. So energy is being released. That energy is being used to plunk a 3rd phosphate group onto an ADP to form ATP. And then the ATP is being hydrolyzed, and in releasing that energy, we are, in this case, building a protein from amino acid components, or subunits or monomers. And this is pretty standard. So ATP, why not just do it directly with glucose then? Looking at this reaction, why not couple glucose with the breakdown of glucose with the building of amino acids? And the answer to that is that ATP is a much smaller price to pay. For every glucose molecule, we can make approximately 36 ATP. And that means that we can power all kinds of reactions with that, as opposed to just breaking down glucose and coupling that with one single reaction. So again, I've used this example in class, but it's like using a $20 bill in one of those vending machines when it doesn't give change, and really you only needed $1. So we don't want to use up a whole bunch of energy that we could use in small chunks. Also, if we were to couple this reaction or these two reactions, the glucose and the protein one, we're going to end up and if we did this sort of as on a regular basis in a living system, we're going to produce way too much heat for that living system to tolerate, because heat is our waste product here, and again, we don't want to spontaneously combust. So the building of ATP, that adding on of that phosphate group, moving from ADP to ATP, can be done in two ways. One of those ways is called substrate level phosphorylation. The other one is oxidative phosphorylation. So these two terms are used to describe two different ways that we can form ATP from ADP. Substrate level phosphorylation occurs in the presence of an enzyme. We've got a nice purple enzyme in our graphic here. And this is an example of substrate level phosphorylation that occurs along the process of cellular respiration. So we are actually going to see this particular reaction in situ, like we're in the pathway, when we get into cellular respiration a little more. And here we're seeing a substrate, which they're calling PEP. And we'll see that's, if I can remember right, phosphoenol phosphate or something like that. So this is PEP from cellular respiration, which is a phosphorylated molecule. You can see a phosphate group there. And in the presence of this enzyme, there's enough energy from all the making and breaking of bonds that occurred so far in the reaction. You can see this is only a 3-carbon molecule, and we started with glucose at the beginning of respiration, so 6-carbon molecule. So enough energy has been liberated that the phosphate group that's here is quite high energy, and there's enough energy to be able to plop it right back onto ADP to form ATP. And then we get the final product in glycolysis, the first set of steps in cellular respiration, and the product we get here is pyruvate and ATP, which we can use to do cellular work. The second way that we can make ATP is by oxidative phosphorylation, and this also involves cellular respiration. And we're going to say this is very cool kind of thing, sort of an more indirect way. We're going to be using what we call the electron transport chain, which is a multi-enzyme complex, which we'll find on the inner mitochondrial membrane, and we all know what a multi-enzyme complex is. And we're going to use that electron transport chain to create a concentration gradient. It's going to be an electrochemical concentration gradient using a proton pump, and we're used to that concept as well. We did Unit 1, we studied what a proton pump might look like, or you know, a active transport involving a protein across a membrane. So we're going to use electrons cascading along the electron transport chain as an energy source to trigger a proton pump. So this is kind of unique. We're not using ATP directly here, which would make it sort of a secondary active transport. So the proton pump is going to be activated by electrons cascading by it, and that's going to pump hydrogen into the space between the inner and the outer mitochondrial membrane. We're going to get a concentration buildup there, electrochemical from the hydrogen ions, and they are going to diffuse back across the membrane into the inner mitochondrial membrane space. And doing so, they're going to pass through a very cool enzyme that's going to phosphorylate ADP back into ATP. So kind of a more indirect way that we're going to be looking at when we look at the electron transport chain, the last part of cellular respiration. And here's an example of a very cool enzyme, ATP synthase, in pink here, embedded in the inner mitochondrial membrane. The hydrogen concentration would be between the inner and the outer mitochondrial membrane. If you're having trouble following this, draw yourself a mitochondria so you know what I'm talking about, which spaces I'm actually talking about. And hydrogen, which was pumped through by the electron transport chain, is found here in concentration and is diffusing back through the ATP synthase and triggers the formation of ATP from ADP and inorganic phosphate, or PI, as is pictured here. Another thing that we had a peek at in Unit 1 are some very important electron carriers that we're going to use in cellular respiration. They're NAD and FAD. And in the first unit, we learned that they were also nucleotides, but they were dinucleotides. And I showed you a couple of pictures where we looked at them. We'll see the pictures again as we move through here. So we're looking at dinucleotides in this case. And they're going to be very important in some redox reactions that are going to be occurring in the process of cellular respiration. And remember, redox meaning reduction oxidation reactions, where electrons are being gained and electrons are being lost. And basically, we're going to see something happening like this, where we're going to have a reducing agent. And remember, a reducing agent is being oxidized, which means it's losing losing electrons. And in losing electrons, it helps something else become oxidized, reduced. So by being oxidized itself, it helps something else become reduced. And if we follow the red arrow here, we'll see our compound B is being reduced. And compound B, which is going to gain some electrons and become reduced, is able to accept those electrons so that A can become oxidized. So if we follow the blue arrow through, we see that B is helping A to become oxidized by grabbing those electrons. So the two things are happening simultaneously. And the whole coupling, again, of this change of electrons is a key concept in cellular respiration. And you'll recall my Leo and Gur, or oil and rig, as some of you guys prefer. And something we didn't talk too much about, but something to keep in mind for the process of cellular respiration, is that the reduced form of a molecule is always at the higher energy level than the oxidized form. The oxidized form has lost electrons, and we can kind of think of that as having lost some energy. And the reduced form has gained some electrons, so is higher in energy. And that's going to, again, be a key concept that we're going to see in the series of redox reactions that are going to occur. All right, this graphic is an excellent little metaphor, I guess, for the way that these carrier proteins, NAD and FAD, work in the process of cellular respiration. They're really transport molecules or electron carriers. And what we're seeing here, sort of in our graphic at the top, we're seeing an empty cab, which is supposed to represent NAD. NAD in the form in which it is low on electrons. So it has lost electrons, it's oxidized. This is the oxidized form of NAD. And we see that there are two electrons looking for a ride here. And in the sort of bottom part of the graphic, we're seeing that there is one hydrogen complete with an electron and a proton, and then one electron that's being sort of grabbed from a hydrogen, and that hydrogen will be become sort of a charged particle floating around there. So we get the reduced version of NAD when we add two electrons and a hydrogen. So this is the reduced form, and they're representing that in the taxi cab by the cab plus the passengers together. The taxi is going to drive the passengers to where they need to go, and then they're going to get out again. So the two electrons are going to be let go in the new area, wherever that might be. And again, it's showing us here in the graphic that these things are going to be used later in the process of cellular respiration. We dump a whole bunch of electrons into something called the electron transport chain later on, and we'll see that in the final stages of cellular respiration. So a little bit of a closer look at NAD, nicotinamide adenine dinucleotide, and you may remember me mentioning that it's this molecule is associated or derived from the vitamin niacin, which you sometimes see on labels of food packets like cereals and so on. And we can see a setup here that looks very much like what we saw with our ATP and the idea of coupled reactions. However, in this case, we're going through a redox reaction rather than hydrolysis and condensation, which we saw with ATP. So there is some exergonic reaction, which is releasing energy in the form of electrons, and the electrons are being picked up by our electron carrier, NAD, to form NADH, which is more energized than the NAD by itself. So remember what we said before, the reduced version is higher in energy than the oxidized version. So we've got now this energized carrier, which is going to carry the electron to power some endergonic reaction, some reaction that requires energy in the form of electrons. So again, acting as an electron carrier to get the electron where we need to go. And as we go through that series, it's showing us here that the net energy available is being compromised by the transfer, but not to the point where it's not useful energy. This graphic is an illustration of NAD, nicotinamide adenine dinucleotide. And again, we can see it's a dinucleotide. I mentioned this before in a PowerPoint when we were looking at nucleic acids. We've got a ribose sugar, we've got our adenine, and we've got our nitrogenous bases, and we've got 2 phosphate groups. So that's basically a dinucleotide. And the flavin adenine dinucleotide, again, we're seeing a graphic similar to one I showed you before. We've got 2 sugar structures here, one in ring form, the one that looks very much like a ribose sugar of some description, so a ring structure. And we've got a nitrogenous base up at the top. Then we've got a couple of phosphate groups. So phosphate group, and then joined to another phosphate group, which is joined to a linear sugar and joined to a nitrogenous base. Okay, we're looking now that we've sort of done a little bit of the, I don't know, the sidebar sort of work, we can have a look at our process of aerobic respiration. And we're going to break it down into 4 different parts, which we're going to highlight first and then talk about in detail. So the first one is glycolysis. I feel like we've been talking about glycolysis a lot, but not specifically. Glycolysis actually occurs in the cytoplasm just outside of the mitochondria. All living critters that carry out metabolic reactions go through glycolysis. So you don't need membranes for this. You don't need oxygen for this. It occurs in the cytoplasm in a multi-enzyme complex that all critters can carry out. It's basically the first 10 steps of aerobic respiration, even though it's anaerobic in nature itself. And in order to get the thing started, we actually have to add a little bit of energy. So we have to have some ATP available to even start the process. But then by the time we finish in those the second half of the process, the 5 energy producing steps, we get back what we use plus a little bit more. So it is overall, we end up with more energy than we started with. This is a sort of summary of the reaction. We're starting with glucose and some ADP, inorganic phosphate, NAD+, and we're going to end up with pyruvate as our final product, some ATP, and some NADH. So we're going from that oxidized version of NAD to the reduced version of NAD. Second step is pyruvate oxidation. This step actually occurs inside of the mitochondria. So pyruvate is a small enough molecule. It migrates across the double membrane of the mitochondria, and this reaction takes place in that middle inner membrane space, or inner mitochondrial space, right in the center of the mitochondria. And again, we see this is another redox reaction, and we're going to be looking at glycolysis in detail. We'll see there's redox reactions occurring there, but we know that because we had the reduced version, or sorry, the oxidized version of NAD that became reduced, had electrons added to it, and we're seeing the same thing here. NAD is becoming NADH, so it's going from the oxidized to the reduced version. We're giving off some carbon dioxide, so for the first time losing carbon dioxide, and we're going to end pyruvate oxidation. I suppose the name of this part is a dead giveaway too, with a molecule called acetyl coenzyme A. So again, occurs in the mitochondrial matrix right in that center part. It's another enzyme, multi-enzyme complex. It says 1-step prep stage, but really there's a couple of sort of steps combined there. And that's the overall for that. So to start, we have pyruvate from glycolysis. We have the oxidized version, NAD+. We've got some coenzyme A, and we end up with an acetyl coenzyme A, NADH, and a couple of hydrogen ions and some carbon dioxide as a waste product. And this acetyl coenzyme A is now ready to enter another stage, the third stage of the process, called the Krebs cycle. And the Krebs cycle also occurs in the mitochondrial matrix, so in that center area. It's a little bit longer here than the prep step or the pyruvate oxidation. So Krebs cycle can also be, or is sometimes also known as, so AKA citric acid cycle. Begins with the acetyl coenzyme A. Now this is actually showing the prep step first, so pyruvate or pyruvic acid here into acetyl coenzyme A. So Krebs actually starts with acetyl coenzyme A, a 2-carbon molecule couples with a 6-carbon molecule that was already there, oxaloacetic acid, here OAA, and forms a 6-carbon molecule called citrate or citric acid, which is where the cycle gets its alternate name, Krebs, from the guy who came up with the series of reactions here. And there's a whole lot of reactions going on here. We can see a lot of redox going on here. So we've got a lot of reducing NAD to NADH, and we've got that other carrier molecule, FAD, being reduced to FADH2. And we're also seeing energy here being given off in the form of ATP, which is being formed by substrate level phosphorylation. So an enzyme catalyzed reaction that doesn't require the electron transport chain. So again, mitochondrial matrix, 8 steps. There's actually more than that, but we tend to condense these steps into 8 steps because a lot of the intermediate steps are very, very quick. And we're going to be decomposing the acetyl coenzyme A, so breaking it down until all we have left from it is carbon dioxide. And we're going to be using a lot of the electrons that are pulled off of the glucose backbone in glycolysis and in prep step, or that's pyruvate oxidation, and in Krebs cycle in the electron transport chain, the final step of cellular respiration. So the electron transport chain is an embedded multi-enzyme complex, and basically this little graphic here is showing us the inner membrane of the mitochondria, and these are all embedded between the inner and the outer space, or the space between the outer membrane and the inner membrane and the mitochondrial matrix. So you can kind of orient yourself, and we can see the membrane here. There's no lipids, but we can see a yellow layer and then another yellow layer here. And there's a whole bunch of different enzyme complexes that are embedded there. And the electron transport chain is literally going to grab all of the electrons that were pulled from glucose, cascade those electrons along, and use it to pump hydrogen from the matrix side of the mitochondria to the inner membrane space, and we're going to have that high hydrogen ion concentration that we were talking about a little bit earlier. Most of the ATP made from glucose is made in the electron transport chain. All right, so we're going to look at these steps, these 4 stages, in a little bit more detail, starting with glycolysis. As mentioned before, glycolysis has 10 steps, and the first 5 steps require oxygen to get them going, and then we get that energy back and then more from the last 5 steps. And some of these I talked about in class already. If we kind of look at our diagram that we have here, we can see that our glucose molecule is being represented here by a linear group of spheres, each representing a carbon. And to begin with, we're using 1 ATP to get things going. And how we use ATP is by coupling. And in this case, we're seeing that the glucose is becoming phosphorylated. And we know which carbon it's going on by the name here. Glucose 6-phosphates, telling us that the phosphate group is going to be attached to carbon number 6 in our glucose molecule. And that's our first stage in glycolysis. And then we're seeing that, and again, it's not the best representation here. I wish we could see this a little bit more linearly, a little bit more like it would be, which is cyclic rather than linearly, but this will do. So we're going from glucose 6-phosphate, and we're going to have an isomerization going on here. So we're actually going to be using, and each place where there's a number here, we have an enzyme involved. So the enzyme at number 2 is an isomerizing enzyme, and it's going to rearrange a little bit. So if you can recall, glucose has 5 carbons in the ring, and fructose has 4 carbons in the ring. So we're having a little bit of a shape change here, and we're actually freeing up sort of another handlebar to hold on to pull this molecule apart, which ultimately is what's going to happen. So we're using another ATP, that's our second ATP, to phosphorylate the other end of the molecule. So we now have a 1,6-phosphorylated fructose-shaped molecule. So our handlebars are in place, and we're going to pull those molecules apart into 2 molecules. One is a ketone, one is an aldehyde, and the aldehyde version is the one that we really want to continue down our pathway with. So we end up with DHAP and G3P, and it's shortened here to GP, but I think you'll see in most of the other material that we're going to be working with, it'll be G3P. And G3P is what we want. So enzyme number 5 is going to help us change isomerize again. It's another isomerase enzyme. It's going to isomerase enzyme is going to change the ketone version into an aldehyde version. And again, you'll remember what that means is the ketone version has a carbonyl group in the middle carbon, and we really want it on the end. So we're going to rearrange things a little bit. Now we're going to have 2 G3P molecules. So again, we started with a 6-carbon molecule. We now have 2 3-carbon molecules, and they're both going to be G3P. Those will continue down a series of steps. And again, we're looking at the product of one thing becomes a substrate for the next enzyme. So for enzyme number 6, G3P is going to enter that enzyme, and we're going to remove some electrons there. So we're going to we're going to make the molecule less energized. We're going to make G3P into a less energy-rich molecule. At the same time, we're going to be adding some inorganic phosphate. So that's phosphate that's freely available in the area where glycolysis is occurring, in the cytoplasm near the mitochondria. And that's sort of a simultaneous step. So that enzyme is able to remove electrons and at the same time attach some inorganic phosphate. And we've had a lot of energy sort of available because of the breaking of the bonds in between step 4 and step 6. And so some of that energy is able to help us plunk that phosphate group on. Again, we're looking at coupling out energy requiring stage with some energy that's available from something that's releasing energy. So transferring bond energy really is what we're doing here. And because the next sort of stage allows us, because we've got this, and I don't know if you can see super clearly, but the phosphate that we just added on, it's showing us here is a high energy bond. So we're seeing that we've got the a sort of squiggly red bond here, supposed to indicate a high energy bond there, again, by utilizing some of the energy when we broke the molecule to do that. So also something you may notice here is everything's got 2s on it. That's because we're working from 2 G3Ps. The next step, we're able to get some of our energy back out again. So we have 2 ADP that are available here, and in this enzyme, we're able to push some phosphate, using some of that energy to push the phosphate back onto ATP. This is substrate level phosphorylation. So we have an enzyme, we have some energy, and we're good to go. And then we do a little bit of rearranging between step 8 and step 9. And something that we're going to see a lot of is water in, water out. And I won't talk too much about that, but it does often have to do with changing the shape or rearranging the molecules. And we end up with this PEP that we looked at before as an example of substrate level phosphorylation. So we know that's what's going to happen in step number 10. We get rid of that other phosphate group, take advantage of some of the energy that's holding it on there. You can see it's a high energy bond. And we are able to make 2 ATP or an ATP there from the ADP. And we end up with 2 pyruvate pyruvate or pyruvic acids at the end. So from glucose at the beginning to pyruvate at the end, 1 6-carbon molecule to 2 3-carbon molecules. And here we've got a bit of a summary. We're splitting things in two. We want a form that's ready to be oxidized once it enters the mitochondria. We know it consumes ATP, but it's going to get us back some ATP. So in this slide, we're, in terms of words, only looking at the first 5 steps. And in the last 5 steps, same graphic, we are creating 2 glyceraldehyde molecules, G3P. And in the end, we end up with 2 pyruvate molecules. We produce 4 ATP by substrate level phosphorylation. So we use 2 ATP to begin with. We need to recoup our losses. So we make back those 2 ATP plus 2 more ATP. So a net gain in glycolysis of only 2 ATP. And there's the in terms of the electrons, we are able to reduce 2 NAD to NADH. And we're going to use that later. We're going to use that energy later in the electron transport chain. So pyruvate oxidation, we've now taken our pyruvate molecule that's in the cytoplasm around the mitochondria, and it's transported into the mitochondrial matrix, so right into the center of the mitochondria. And it's getting prepared to go into KREB'S, so it's often called the prep step. We're going to have our pyruvate. We can see the little blue portion here. Once it's migrated across the mitochondrial matrix, the first enzyme is going to, or the first step of pyruvate oxidation, we remove carbon dioxide. And we'll see along the rest of the steps in KREB'S, every time we remove carbon dioxide, we reduce a molecule. So we reduce NAD to NADH, higher energy molecule. We also have this coenzyme A that's freely found inside the mitochondria that's going to become attached, and it's kind of like your ticket in. It's like your, your special backstage pass to get into the KREB'S cycle. And what we end up with is a molecule we call acetyl coenzyme A. So first carbon group removed. We have 2 carbons now, 2 carbon for each molecule that came from glycolysis. And we have our reduced NADH. Again, we're seeing here that coenzyme A is another vitamin derivative, kind of like we saw our carrier molecules were vitamin derivatives, FAD and NAD. And I think I said the NAD from niacin, but the FAD is derived from riboflavin. Acetyl coenzyme A that we've just created in that prep step is a very versatile molecule, called here pivotal molecule for cellular metabolism. And it's very versatile in that it can have things added to it or taken away from it or slightly changed to allow it to become part of the pathway for lots of different things. So it can be used to create fatty acids, it can be used to make ketones, it can be used to make cholesterol. We can continue through and finish with our cellular respiration. It's a good entry point for amino acid or protein catabolism, so the breakdown of protein. It's good intermediate for the formation of triglycerides, for phospholipids, and so on. So very important intermediate reaction, or intermediate molecule, and can be used in as a part of many different reactions. And in each case, basically, we've got something that's reversible. So this allows us, for instance, to be able to break down amino acids to be used to store as fat, for instance, or allows us to use fat stored to build certain proteins or certain amino acids that we can use to make proteins. KREB'S cycle is our next phase, and we can see our 8 steps here. Sorry for that. KREB'S cycle is going to occur twice per glucose molecule, because remember, for each glucose molecule, we made 2 pyruvate, which in turn were converted into 2 acetyl coenzyme A. So 2 turns of the cycle for every 1 original glucose molecule. So for each turn, we have a 2-carbon acetyl coenzyme A enter, and you see as soon as we go in here, coenzyme A is released to be reused in the prep step. And all of the carbon that comes in is going to leave as carbon dioxide. So if we look around this circle, we'll see pretty quickly that we had 2 carbon come in, but on the right-hand side, we're going to have 2 carbon dioxide come back out again, along with some other things that are that's going to happen there. So acetyl coenzyme A couples with this oxaloacetic acid or oxaloacetate to form citrate. That's the beginning step. And again, each one of these is catalyzed by an enzyme. We see we've got some isomerization going on here, and we go from citrate to isocitrate. 6 carbon, 6 carbon. Remember, we had 4 carbon oxaloacetic acid and 2 carbon acetyl coenzyme A to make a 4 carbon citrate, or sorry, a 6 carbon citrate, 6 carbon isocitrate, and then we start to get rid of those carbons that we brought in again. So part of a cycle means that we have to end up with the stuff we started with. So we have to end up with the we have to end up with the oxaloacetate or oxaloacetic acid that we started with. So we're going to get rid of our first carbon dioxide, and at the same time, as I mentioned before, coupled with carbon dioxide being released, we have a reduction reaction occurring there. So our isocitrate is becoming oxidized, our NAD is reduced, and we're able to use that energy later in the electron transport chain. We now have a 5-carbon molecule, which is very quickly oxidized again. NAD is reduced, carbon is released. So now we're down to 4 carbon, but there's still, from all that bond breaking and shifting, we've got a lot of energy still left that we can use. So we've got a series of reactions to get back to oxaloacetate, during which time we're able to suck a little more energy out there by first producing ATP by substrate level phosphorylation, sort of at the bottom of the visual cycle here. We are able to reduce FAD to FADH, and again, remember these are redox reactions, so every time we're reducing something, we're oxidizing. So succinate is oxidized to fumarate, and then malate is going to be oxidized back into oxaloacetic acid, so low energy molecule. Remember, the reduced molecule is going to be higher in energy. So for each 1 turn of the Krebs cycle, we've made 3 NADHs, 1 FADH2, and 1 ATP. So that's a lot of potential energy there. Okay. Some of the energy, as I mentioned, is used to create ATP. And for each glucose molecule, remember, we're producing 2 acetyl coenzyme A, and therefore there's 2 turns of the glucose, or 2 turns of the Krebs cycle for each 1 glucose molecule. There's another visualization of the process. All right, and on to the last phase of cellular respiration, called the electron transport chain. And here we see another visualization of the inner mitochondrial membrane. And we will basically, to orient yourself, the top of the graphic is the inner membrane space between the inner mitochondrial membrane and the outer mitochondrial membrane, and the bottom space is the mitochondrial matrix. And again, we understand that this is a multi-enzyme complex, that in this case it's embedded in that inner mitochondrial membrane. And we're going to use this to harness the power from the electron, so electron power. And we're going to use that electron power to create an electrochemical gradient, which ultimately is going to allow hydrogen ions to diffuse back through a special enzyme called ATP synthase, and for ATP to be produced. And we're going to produce a lot of ATP this way. So another visualization of the electron transport chain, this time a little more simple, but same idea. We're seeing a concentration of hydrogen ions at the top, and we're seeing this ATP synthase in purple that's going to allow us to make ATP. In the writing here telling us that each component of the electron transport chain is a little more electronegative than the previous. And you'll remember about electronegativity from Unit 1. The more electronegative something is, the more able it is to grab electrons. So that's going to set us up with a cascade of electrons. So the electrons are going to be pulled a little bit harder. It's kind of like having a series of people in a I don't know, like a tug of war chain or something like that, where the first is the weakest and the one at the end is the strongest, kind of like your anchor in a tug of war. So the guy at the end is going to have the strongest hold on those electrons. And our final electron acceptor, and the first time that we've actually talked about oxygen, even though we're talking about respiration here, cellular respiration, oxygen is actually our final electron acceptor. So it is the most electronegative of all of the, compared to the components of the electron transport chain. And we are going to be dumping our electrons ultimately onto oxygen, and then we're going to form water from that, and that's going to be given off as a waste product. So when we think about oxygen and the role it plays in respiration, really that's it. It's our final electron acceptor, and without it, the electrons will not be pulled through the electron transport chain. We will not be able to create a concentration gradient, and we will not be able to make any of our ATP using the electron transport chain. Okay, so a lot of writing here. I like this graphic. Have a look at what you're actually looking at. So along the y-axis, we're seeing chemical potential energy. So the things at the top have the highest energy, and on the bottom, we're seeing time. So these are process reactions that are going to occur, redox reactions, and it's a whole series of them that's going to occur as electrons are being passed into the electron transport chain and then between all the components of the electron transport chain. And remember, as we're going along here in time, we're moving toward things that are more and more electronegative. So from our carrier molecule, remember our taxi cab that's full of the passengers. Our passengers are getting out here, and the electrons are going to be passed on from NADH to the first component or first complex of the electron transport chain, cytochrome B complex. And that's going to be more electronegative than the NADH, and it's going to have a grab a hold of those electrons, and that complex is called NADH coenzyme Q reductase. So makes sense, right? We're reducing, we're gaining electrons, or the complex is gaining electrons from the NADH becoming reduced. What's happening to the NADH? It's lost its passengers, it's back to its oxidized form, and it can be reused by the components of cellular respiration. So it's ready to be reduced, if you like, pick up electrons and bring them over here. So the cytochrome B complex has those electrons for a very brief period of time, but then the cytochrome C complex is actually got a higher electronegativity, is able to pull those electrons away. So cytochrome B is going to give up the electrons and become oxidized, and cytochrome C is going to pick them up and become reduced. And then cytochrome C is going to drop those electrons off to cytochrome A, which is more electronegative, which now becomes reduced, and so on. So you can see it's a whole cascading series of redox reactions. Cytochrome A complex is going to be able to drop those electrons onto oxygen to form water. So that's basically what's happening. We've got this whole, as we're describing it here in the words, highly exergonic process where we've got little steps and we're able to release energy. And what's not being shown on here is all the hydrogen pumps that are being activated as these electrons actually cascade through. So again, if we look at this blue graphic over here, we can see that we've got electrons being passed through. They're being signified here, 2 electrons, 2 electrons. And we see 3 hydrogen ion pumps. So in complex 1, we have a hydrogen ion pump, and we can see in the red arrows that hydrogen is being pumped through to the inner membrane space. There's another hydrogen pump in the cytochrome B, cytochrome C complex, and then another one in the cytochrome C to cytochrome A complex. So 3 separate hydrogen pumps triggered by the electrons that were dropped there by NAD that started that series, that cascade of electrons through the electron transport chain. At the end of cytochrome A, we're seeing the electrons being dumped onto oxygen to form our final waste product, which is water. So that's all well and good. However, we do have another electron carrier. It's that FAD molecule. We only make 1 of those per turn of the Krebs cycle, but it still has some potential energy there. We're also going to see that it's going to be able to grab the energy from the NADH that was produced outside of the mitochondria. So FADH2 is a great molecule for carrying electrons, but it's not as high energy as NADH. The reason for that, or one of the reasons for that, is that the docking station, if you will, the place at which FADH2 is able to unload its electrons in the electron transport chain, comes after the first pump. So again, have a look at where the arrows are going in here for FADH2, where it's dropping its electrons in the sort of pink flesh colored area here. And it's going to be joining us after cytochrome, the cytochrome B, or sorry, just before the cytochrome B complex, just after that first part of the electron transport chain. It's missing the first pump. It does not trigger that pump. Instead, it triggers pump 2 and pump 3. So not as much of an impact on the hydrogen ion concentration, so it does not contribute as much energy to form ATP. In fact, it yields 1/3 less energy than NADH, as you probably figured out from the graphic here. Here's maybe a better overall look at what sort of the whole mitochondria. We see the matrix in the middle and the inner membrane space here in sort of a yellowy color. Something that's good to note as well is that there's multiple electron transport chains in this membrane, hundreds and hundreds of them, in fact, and same with the ATP synthase enzymes, lots and lots and lots of them occurring at the same time. So I was mentioning earlier that the NADH from the cytoplasm, from the glycolysis reaction, you remember there was some NADH that was made there. The NADH can't move into the mitochondrial matrix. It's not able to migrate across the two-membrane system. But the electrons are shuttled onto an FAD to become FADH2. And those electrons, therefore, also will get dumped into the electron transport chain in the same location and will yield 1/3 less energy. Okay, so even though we get some energy from that in glycolysis, not much. If we were a prokaryotic cell, the NADH that was liberated, or that was, created, would not be able to enter the electron transport chain because there wouldn't be one. And we'll look at the impact of that in another in a different lecture. So keep in mind that we also have to have some strategy to deal with that if we are running out of oxygen, say if we're into sort of oxygen deprivation or oxygen starvation in our muscles, if we're running or something like that, and we've used up all the available oxygen. We still carry out glycolysis, we still make that NADH, but we have nowhere to dump the electrons. So we're going to deal with what happens to those in another lecture. All right, so often in the process of cellular respiration, you hear the term chemiosmosis. And what we're referring to there, again, chemi meaning chemical, osmosis meaning, well, osmosis, moving from a high concentration to a low concentration. So with these proton pumps that we've got triggered by the electron cascade through the electron transport chain, we have hydrogen ions being pumped in to form a concentration gradient in the inner membrane space. Then we are going to have a chemiosmotic experience. They are, at least. Hydrogen is going to diffuse back through ATP synthase to form ATP. So the chemiosmotic episode or experience or situation is basically that hydrogen ion migrating back through the ATP synthase to form ATP. And we can see it in this diagram. We've got the electron transport chain in sort of big parentheses, and then we've got chemiosmosis after that. So the production of ATP from the diffusion of hydrogen ion through a protein carrier, the ATP synthase enzyme in the membrane. The whole thing together is allowing us to do that oxidative phosphorylation that we mentioned earlier. This is the formation of ATP using the cascading electrons to form an electron, hydrogen ion concentration, which will allow them to diffuse through ATP synthase. The only way to get back through that membrane is this enzyme and form ATP. ATP synthase is quite a complex, very complex enzyme embedded in the inner mitochondrial membrane. And it's pictured in lots of different ways. This is quite a complex sort of look to it. And we're always learning, or presently at least, learning new things about how exactly this works. Again, for our purposes, hydrogen ion is going to be able to move through that, and only through that. Can't go through the bilayer, as we know, and there's no other protein available for it to go through, so must diffuse back through that enzyme, and in doing so will trigger the coupling of ADP and inorganic phosphate to form ATP. All right, so let's look at our totals for cellular respiration. From glycolysis, which occurred in the cytoplasm, we had a net gain of 2 ATP. Remember, we used 2 ATP. We produced 4 for a net gain of 2 ATP, and that's signaled here at the top here, 2 ATP. That's by substrate level phosphorylation. We also yielded 2 NADHs. So we oxidized the glucose components, and we reduced NAD to NADH. Normally those would be able to yield 3 ATP each, but because you remember we said they can't migrate across the membrane, so their electrons are shuttled onto FADH2, which can only produce 2 ATP per molecule. So same thing here, we get our 4 ATP from that. Moving the pyruvate then, of course, moves into the mitochondrial matrix, and in the mitochondrial matrix we go through the prep step. Pyruvate oxidation, where we yield 2 more NADHs, this time inside the mitochondrial matrix. So we know that those electrons will be dropped off at the beginning of the electron transport chain, and will yield 6 ATP in total, because it's going to trigger all 3 pumps and allow enough hydrogen ions across into our concentration gradient that we can yield 3 ATP each, or 6 in total. Then after our prep step, our pyruvate oxidation, acetyl coenzyme A is going to enter the Krebs cycle. And in the Krebs cycle, we're going to produce 2 ATP by substrate level phosphorylation. If you remember our diagram, it was sort of down at the bottom where we would expect 6 o'clock to be. And we also are going to be able to reduce a lot of NAD to NADH. In fact, 6 of them per 2 turns. So we're looking at a complete glucose here. 2 turns of the cycle. Those 6 NADHs have enough electrons that we're able to pump 3 hydrogen, or we're able to trigger all 3 hydrogen pumps and yield 18 ATP for those electrons. And we also are going to reduce 2 FAD to FADH2. Each of those enter a little bit later in the cycle, as you'll recall, able to trigger 2 hydrogen pumps and yield 4 ATP for a total net gain of 36 ATP per glucose molecule. If you find any of that confusing, make sure you have some questions when you come into class.